Good morning, friends. Today we are going to discuss about the basic genetic concepts. Okay, first of all, we will discuss about the definition of genetics. So, what is genetics? Genetics is the study of heredity and variation. So, first, what is heredity? Heredity means the transfer of characters from one generation to another generation. That means the characters which are transferred from parents to their children. That means the characters which are present in their parents are also seen in the children. That means in their offsprings. So that is called heredity. And what about the variation? The characters which are observed in offsprings but not in parents. That means the characters that are not present in the parents but that are produced newly or synthesized newly or expressed newly in the children's or offsprings. That is called variation. So the characters which are transferred from one generation to another generation and the characters which are observed in offsprings but not in parents, the study of these two factors is called genetics. So who laid the foundation for genetics? Means the foundation of genetics was laid by Gregor Johann Mendel, G.J. Mendel. So he is called father of genetics. He is called father of genetics in the year 1866. But he proposed two laws, law of independent assortment and law of segregation. Because of his laws related to mathematics at the time in 1866, these are not acceptable by all the scientists. Later, three scientists, Hugo de Vries, Correns and Chermark, they started some experiments and they found the Mendel's experiment they found, and they find that Mendel already told about the genes. According to Mendel, genes are nothing but the factors which are written by Mendel's are nothing but genes. Okay, so Mendel's findings came into light in the year 1900. So after his death, the Mendel's findings, the research papers of Mendel's came into light in the year 1900. So they decided that genetics was born in the year 1900. And whenever we discussing about the term genetics was coined by Bateson in the year 1905. Okay, so the term genetics is derived from the word Greek language. So okay, a small discussion. Whenever in the competitive exams you will be asked like the term pomology or whatever it may be, logo or logi or logos, if you will get definitely this word will be derived from Latin language. So please remember this. Okay, and what is the meaning of genetics? To grow or to become. So the as the characters here already we have discussed that genetic deals with the characters. So the characters are generally governed by the genes. So the characters are governed by the genes. So genetics is the study of structure, composition and function of genes. That means what is the structure of gene and what is present in gene, that is composition, and how the genes will function to express any character. So all these terms also we will discuss under the genetics. Okay, so here whenever they are asked like what is genetics, we can say that genetics is the study of heredity and variation. Heredity means the transfer of characters from one generation to another generation, and variation is the characters which are observed in the offsprings but not in parents. And we can also say that as characters are governed by genes, so the genetics is the study of structure, composition and function of genes. Okay, whenever we come to the discussion of branches of genetics. We are having a lot of branches. That means plant genetics, microbial genetics, animal genetics, molecular genetics, biochemical genetics, population genetics, radiation genetics, eugenics, Mendelian genetics, genomics, euphenics, cytogenetics, and quantitative genetics. Okay, first of all we will discuss about the plant genetics. So here in the plant genetics, it is the branch of genetics which deals with the inheritance of characters in various plant species. That means the characters which are transferred from one plant species to another plant species, and the variations that are occurred in offspring plant species which are not observed in their parent plant species. So these terms, these will be studied under the plant genetics. And whenever we are discussing about the microbial genetics, it deals with the inheritance of characters or traits in microorganisms like bacteria, fungi, and virus. So the characters which are transferred from one microorganism to another microorganism, the microorganism will be anything that may be bacteria or fungus or virus, whatever it may be. The genetics which deals with the inheritance of characters or traits in microorganisms like bacteria, fungi, and virus, that is called microbial genetics. So animal genetics, next thing. So it deals with the inheritance of characters or traits in animals. That means the characters which are transferred from one animal to another animal, that is called animal genetics. Molecular genetics, it deals with the structure, composition, function, and replication of chromosomes and genes representing genetic material. What is the genetic material? DNA and RNA. So the structure of nucleic acids, that is DNA, RNA, that means structure of DNA, structure of RNA, and the composition of DNA and RNA, and function of chromosomes, and how the chromosomes will behave, and the replication of chromosomes during the cell division, and genes, and how the genes are placed on the chromosomes, and how these genes are functioning along with the chromosomes, and representing genetic material, that is DNA and RNA. All these stuff we will study under the molecular genetics. And biochemical genetics, it deals with the role of genes in controlling biochemical pathways in the organisms. That means how these genes are controlling the organism. That means how these genes are involved in the controlling of biochemical pathways, that is metabolical pathways, how the genes are playing their role in metabolism of an organism, that we will study under biochemical genetics. And when we come to population genetics, it is the study of genes and genotypes, that means frequencies of genes and genotypes in a population, as well as with various agencies which tend to alter gene frequencies in a population, leading to evolutionary changes. That means in a population the genes and genotype frequencies are studied, and the factors which will change the frequencies in a population to evolutionary changes. So that also we will study under this population genetics. And radiation genetics, it deals with the effect of various types of radiations on chromosomes and genes. That means the radiation effects which which effects of various types of radiations on chromosomes and genes. And next we come to eugenics. Eugenics means it deals with the application of genetic principles for the betterment of human race. That means using the principles of genetics and the science of genetics for the improvement of human race, for the benefits of human. Okay, that we will study under eugenics. And Mendelian genetics, it deals with the inheritance of qualitative characters or oligogenic characters which display discontinuous variation. So this we will discuss in Mendel's law. So okay, no problem. Next, when we come to quantitative genetics. Quantitative genetics, it deals with the inheritance of polygenic or quantitative characters which display continuous variation. Generally, qualitative characters or oligogenic characters show discontinuous variation, and quantitative genetics show continuous variation, and they are polygenic characters. This also we will study in the higher classes. This branch of genetics is also known as biometrical genetics or statistical genetics or mathematical genetics. So qualitative genetics is also called biometrical genetics, or statistical genetics or mathematical genetics. Okay, next, cytogenetics. It is the study of cyto means cell, cytology. So okay, the cytology term is derived from Latin. Already we have discussed here logi or logos. When we see these terms in any term, these words in any term, that is definitely derived from logos. That means to study. Logos means to study. Okay, it is the study of cytology and genetics. Okay, next, eugenics. It deals with the control of hereditary diseases, especially inborn errors of metabolism. It deals with the control of hereditary diseases. That means the diseases which are present in parents also observed in their children. That means the hereditary diseases means the diseases which are transferred from one generation to another generation. So the study of that hereditary diseases is called eugenics. Okay, next, genomics. Genomics is nothing but study of structure and function of genome of an organism. Okay, so these are the branches of genetics. Okay, next topic is forward genetics and reverse genetics. Okay, we will discuss the difference between the forward genetics and reverse genetics. In the forward genetics, first the phenotype is known. That means the character which is observed with our naked eye can be first identified, and after that the gene involved to produce that phenotype is identified or examined. That is, the examination of an genetic cause of an altered or abnormal phenotype introduced by a chemical mutagenesis or mutation by radiation. That means if we observe a plant is a plant is having very small leaves. For example, a plant is having a very small leaves. The character small leaf, that means small leaf is coded by one of the gene. So here first we have find out the phenotype. That means the small leaf, that is the phenotype we have. First we will find out that, and we will search for the gene which is involved in the production of that phenotype. That is phenotype to genotype. What is the meaning of genotype? The genetic makeup. Phenotype is nothing but the genetic makeup. And forward genetic includes classical genetics. Okay, reverse genetics. Here the total reverse thing will happen. First the gene is identified here. The first gene is identified, and then the phenotype produced by the effect of the gene is identified. That means the gene is identified first, and the gene will produce a phenotype. The genotype will produce a phenotype that is identified next. So genotype to phenotype. First we will identify the genetic makeup of a gene, and after that we will discover the phenotype. That is, the particular gene is altered and the phenotype is examined. This includes the genomics. Okay, thank you.